אז בוקר טוב לכולם. Good morning. We have with us Professor Paul Schreiner. I'll introduce him in Hebrew. אז בוקר טוב לכולם שוב, אנחנו מקליטים את ההרצאה, תקבלו אותה וגם את בסוף יחלוק איתנו גם את ההרצאה ב-PDF ונשלח. אז פרופסור פול שריינר, מי שלא מכיר, הוא עובד ב-USDA, זה המחלקה לחקלאות, נקרא לזה, אמריקאית, יושב באורגון, ומי שמכיר אותו, וקצת יצא לו לעשות גוגל, ואפשר לראות גם באתר של ה-USDA, חוקר הזנה בגפן, ובכלל פיזיולוגיה, אבל הזנה בגפן הרבה מאוד מאוד שנים, יש המון פרסומים בנושא, ויש לנו עכשיו את השעתיים הקרובות לבלות איתו, הוא יעביר את ההרצאה, אנחנו מציעים, כמו שנתיב אמר, שתשאלו שאלות בצ'אט, אלא אם כן הוא יעצור ויפתח את זה לשאלות, ואנחנו, אם מישהו רוצה לכתוב בעברית, אפשר גם לכתוב בעברית השאלות, אני יכולה לתרגם, ונראה איך אנחנו מסתדרים עם זה, כי כמו שנאמר פה, יש הרבה משתתפים היום. So, Paul, if you would like to introduce yourself, and you can start the presentation.
Okay, thank you, Noah, and thank you for the introduction. I have no idea what you said.
All good things.
Sorry, I don't speak any Hebrew, so yes, so I'm Paul Schreiner. I'm a plant physiologist, and I'm in Corvallis, Oregon, which is on the rainy side of the Cascade Mountains on the west coast of the United States. Before I get into my talk, which I'm going to talk about nutrition in wine grapes, I want to just give a very, very brief introduction of what my lab does. For some reason, it doesn't want to advance. Okay, there we go. So I basically do 4 different things in my lab, or I work in 4 different areas. They're all somewhat related. The first is optimizing nutrient inputs based on vine physiology and fruit quality. I work only in wine grapes because that's what we grow in Oregon and in Washington, where I'm from. I do not work on other wine grape or other grape commodities like table grapes or raisins. That's pretty much all in California. Okay, so beyond the wine grape nutrition, I also look at impact of management practices on the plants themselves, on mycorrhizal fungi, and nutrients and vine performance. And my background is in mycorrhizal fungi. That's what I got my PhD in. So I always try to do at least a little bit of work on mycorrhizal fungi. Another area that we have been working in is characterizing the fungi that are there, what role they play, what functions they play, and looking at the community. And then finally, we like to look at interactions between roots, mycorrhizal fungi, and other biota that are in the system. In the past, I've looked at calambula, and I've looked at some other soil organisms. But presently, the work that we're doing is focusing on nematodes. I won't talk about any of that today, the nematode stuff, but this is an overview of what I'm going to try to cover. And depending on how things go, I may cut short a few slides and just skip over some stuff. But I'm going to send you this PowerPoint, or I'll send it to Noah to forward to everybody. So today's talk, I'm going to give just some background on vine nutrition. I'm going to talk a little bit about tissue testing. Then I'm going to talk about the time of nutrient uptake, basically when are vines acquiring nutrients from soil, which obviously would have an influence on when you'd want to fertilize vines or fertigate, depending on how you're managing your nutrient inputs. The third part, I'm going to talk more specifically about how much nitrogen, phosphorus, and potassium do vines need, using the example of Pinot Noir, which is we grow here in Western Oregon. And then finally, I'll wrap up with understanding and managing mycorrhizal fungi in vineyards. So just a little quick review here on what we typically think about in terms of nutrition. These are the most common elements that are in grapevines. The top three, of course, carbon, hydrogen, and oxygen, come either from air or from water. When we talk about nutrition, we're usually talking about the mineral elements, which are all the rest following those. So nitrogen, phosphorus, potassium, calcium, magnesium, sulfur, those are all the macros. And then the micros, iron, zinc, manganese, copper, boron, and molybdenum. Today, I'm going to mainly focus on N, P, and K. And a little background on nitrogen. Of course, nitrogen is in a lot of really key molecules or compounds that are in all living organisms, including chlorophyll. Nitrogen is involved in every pathway in life, basically, right? Because the biochemistry of life is performed by proteins or enzymes. Nitrogen is often the most limiting nutrient in agriculture. Not always the case for viticulture, I will say. However, even if it's not limiting, it might still be the most important nutrient to get right. Sort of like the three bears story. I don't know if you guys have that story, but you know, mama bear, papa bear, and baby bear with the porridge being too hot, too cold, or just right. In wine grapes, especially, you want nitrogen to be just right. And that's because nitrogen drives a lot of key things. Vegetative growth in particular, that's the big thing. Of course, it also influences yan in the berries, or yeast assimilable nitrogen, which, depending on the winemaker that you are working with or know, or if you're the winemaker yourself out there, you might have a very different idea about what yan should be. But the other thing, of course, is nitrogen influences fruit exposure, and it also influences the microclimate around the fruit. So of course it has a huge impact on disease as well. So this is the thing that you don't want to do with nitrogen, is have such vigorous vines that you're hedging off all the stuff you can see laying in the alleyway here. And these extremely green vines that are very, very full. Almost every place that grows grapevines, well, especially in the New World. Okay, so I'm coming from the New World. A big mistake a lot of people make initially is they put on too much nutrients, they put on too much water, and they get this. You don't want that. Of course, this is a picture from one of my pot and pot studies, which I will refer to quite a few times today. You probably don't want to be quite this weak. On the other hand, it's a little bit too weak. So the 5 vines in this front panel here that you can see are my lowest nitrogen supply from an experiment we did. And this is a little bit too little growth. However, if I had to choose between that and that, I would still choose this. You might have a slight yield reduction, but your fruit quality is going to be way better. Okay, so a little review about phosphorus and potassium. So phosphorus is really important because it's in cell membranes, nucleic acids, and it's also part of the ATP, which is the energy cycle of the cell. Adequate P is needed for most things in plants. Typically, the things we think about being most sensitive are root growth and flowering, as far as when potassium is getting too low. Typically, phosphorus. Phosphorus is the second most limiting nutrient in agriculture, typically. Now moving on to K. K is unique because it's not covalently bound. It just exists as an ion. But it plays a really fundamental role because it's probably the most, or one of the most important ions in terms of osmoregulation, charge balance, and it influences a lot of things, including protein synthesis as a result of all these things. K, typically thought of as the third most nutrient limiting mineral for agriculture, potassium. The key thing with wine grapes especially is that potassium influences berry pH and hence wine pH, and that has a big impact on wine quality. And root uptake of both phosphorus and potassium is mainly driven by diffusion. And I'm just going to give a little review here about roots and nutrient uptake. These are the three major pathways that nutrients get to the surface of the root. One, the root grows to the nutrient or basically contacts the nutrient. That's called interception. The second thing is through bulk flow. So these are nutrients dissolved in the soil water, which are moving because of transpiration through the system and pulled in to the root in that manner. And then the third way is through this idea of diffusion. And diffusion is where phosphorus and potassium are mainly taken up. And indeed, when you look at some, this is kind of an old slide, but it's still true today, both modeling and scientific data, you can see the nitrogen primarily was moving to the plant through bulk flow because it was taken up as nitrate, and nitrate's highly soluble. However, both phosphorus and potassium, the primary route of getting to the root was through diffusion. And I'm not going to spend time on this, but of course, once the nutrient's at the surface of the root, it also has to be taken up into the cells. And of course, that means crossing the lipid membrane. Sometimes you'll hear people say, oh, this ion is taken up passively, or this ion is taken up actively. But really, you have to think of it, or in a practical sense, I think of it, it's active no matter what, because the plant has to build the membrane and all the proteins that perform these functions. And here's an example of a structure, model structure of what it looks like if you were looking at one of these membrane-bound transporters from above. You can see there's basically a hole right through the middle of that. And the size of that hole is what dictates what ions can go into that or out of that. And then some of these transporters have gates that either are open or shut. So just a little background now on nutrient uptake by roots. I've covered some of this. Of course, the first thing is it's an active process mediated by carriers that are present in cell membranes. I just showed that picture. Roots concentrate ions a huge amount compared to how much their concentration might be in the soil. Over 1,000-fold increase in concentration between the soil nutrient level and what's inside the root cells. And ion uptake is driven by this electrochemical gradient. I'm not going to talk about that any further. That's a little above maybe where we need to be today. But because it's an active process, nutrient uptake can be limited by certain environmental conditions that limit the biology, such as temperature, moisture, pH, oxygen, lots of factors. Now, when we talk about a perennial plant like grapevines, the fine roots, which are the ones at the very tip, which almost no one ever sees, they play the big role in terms of nutrient uptake compared to the woody roots, which is the main structure of the whole root system. And indeed, mycorrhizal fungi play a significant role in the uptake of many nutrients, particularly phosphorus and other less mobile nutrients. So before I move on to talk about tissue tests, I just wanted to say a couple words about nutrient testing as a method for nutrient management in vineyards. I'm not a big fan of this, just because as a plant physiologist, if I can measure what's in the plant, it just makes more sense to measure what's in the plant. Measuring what's in the soil gives you potential, but it doesn't actually tell you what the plant's getting. And indeed, we looked at this issue. And so this is data from a survey I did here in Western Oregon when I first started working on grapes, where we collected data from 29 different vineyards. We extracted nutrients from the soil, and then we also measured nutrients in the leaves. And all the nutrients are listed here. Of all the nutrients we looked at, only one was significantly correlated between what was in the soil and what was in the plant, and that was nitrogen. But it was weak. That's only accounting for 18% of the variation. So the short answer to this question is no. Soil tests are not a very good predictor of what's in the vines themselves. And indeed, I have to bring this quote up. So I was 3 years old when Cook wrote this, and this Cook was a nutrition guy that worked in California. Basically, you can read that quote. The bottom line is soil tests were mainly, how do I say it correctly? Mainly developed for annual plants, one thing. And so a lot of the nutrient tests were only to predict nutrients in a very short time window where annual plants would take those nutrients up. When you try to apply that to a perennial system, it's just not as good. And it's obvious because grapevines are there for a long time. They have a longer period of activity that they can take up nutrients. And our ability to extract something is just not as good or not as accurate as, it just can't mimic the real world. All right, so I want to give now moving on to tissue testing. So, pardon me, when I say tissue testing, I'm talking about what's in the plant. I don't know, and maybe someone can tell me in the chat box, and I'll look at it later, what kind of tissue tests you're doing in Israel. I could not find that information in the time I had to prepare for this talk. But the history of the tissue tests, based on what I knew, is given here. And we cover this in a paper I published recently in 2017. I guess that's not so recent. But basically, the French were the first that started doing this, as far as I could find in the literature. And they were using whole leaf blades to assess mineral nutrition of the vines. As a result, leaf blades are still used today in Europe as the primary tissue that's used to test for nutrition. In the United States, especially well, it was mainly in California, work was very focused on using petioles for nitrogen and potassium mainly. And it got picked up by New York, also got picked up by Australia, and then petioles became kind of the chosen thing in those regions. And then across the US, petioles became the tissue of choice. However, as I point out here, a number of us have questioned why you would use the petiole, because the leaf is what does the work. The petiole is just a little stalk holding the leaf up. So we look at this, and I'm going to talk a little bit about this paper where we compared leaf blade versus petioles as predictors of nitrogen, phosphorus, and potassium status in Pinot Noir. So what we did, it was really very basic, was we compared leaf blades to petioles at standard time of bloom, or 50% cap fall, and then also at veraison, or 50% color change. Leaves and petioles were collected, separated, and dried, normal procedure. And then we used the raw data from our experimental units. And then we looked at how well either leaf blade or petiole nitrogen or other nutrients predicted vine responses. And so the data that we used for this, I'll talk more about later, and you'll see why I use this data. But it came from this pot and pot system. I showed a picture of it earlier already where those vines were kind of weak, that were low nitrogen. So we use this pot and pot system where we were basically carefully controlling what the vine could get access to. And so we had 5 rates of N, and we also had 4 applications, and then we have 4 years of data. And then we also added data that we had from commercial vineyards, from just what I had historically in my collection of data from various trials we've done over the years. And I'm just going to talk about nitrogen. So here is, for example, prune weights prune weights on the y-axis versus leaf or petiole nitrogen on the x-axis. Hopefully you can see this. It's really pretty clear that the R squared for leaf nitrogen or leaf blade nitrogen is almost 2 times better than it is for petiole. This is for predicting prune weights. If we look at yield, a very similar situation. If we look then at YAN, which is something that ought to be even more closely tied to vine nitrogen status, again, we see the same thing. It's about 2 times better in terms of model fit when we look at leaf blade as opposed to petiole. Now, the only data I had from the field from all these other studies that I could look at was YAN. And so we looked at that, just the must-YAN aspect from all different kinds of fields around Oregon. And here you can see the relationships are not as good as from my pot and pot study because obviously it's not as well controlled. But again, the same story, leaf blade N was a far better predictor of YAN than was petiole N. So basically, the summary of that work is that leaf blade always outperformed petiole. And basically, the wider variance in the petiole was a lot of times attributed to the year, which I'm not going to take time to show you that data today, but sometimes, for example, we just have higher petiole N years than other years, and I don't know exactly what controls that. Leaf blade tends to be more consistent from year to year. Also, we looked at phosphorus and potassium in this paper as well. And when we looked at those nutrients, we didn't see a significantly better relationship with leaf blade versus petiole. However, some subsequent work that I've done, we found leaf blade was better. So to me, this kind of made sense. Of course, you would say, well, gee, it totally agrees with what your initial idea was. So how do we know you're not making up the data? Well, you've got to take my word for it. So moving on to a case study, which is going to exemplify this whole issue in another way. And so this is a vineyard we were working in 2013, and towards veraison, the onset of ripening, we started to see these symptoms in the vineyard on the leaves. Now, most of you may know what those symptoms are probably due to. If you have some experience in viticulture and have some experience with different kinds of nutrient deficiencies, but there's really 2 nutrients that are most likely. And so we looked at this, and the reason I'm sharing this is because this is a way I recommend to growers to try to diagnose when they have problems. Make sure you take a sample from the symptomatic leaves, but also collect a sample from the same vineyard, try to find the same age leaves on nearby vines, further down the row or in another row, but of similar age that are non-symptomatic, and submit them both. That way you get the answer. If you only submit the samples that have symptoms, you can't always tell for sure from the tissue test that you're going to get the right answer. So I imagine many of you probably would have guessed the 2 likely nutrients that give these symptoms are magnesium deficiency and phosphorus deficiency. This marginal reddening, and then it also tends to be interveinal. Well, you can see from the results from our tissue analysis, clearly the ones with symptoms were deficient in magnesium. They were fine for phosphorus. An important point though is, had we used the petiole, here's the petiole data with magnesium, we would not have been able to tell that magnesium was the problem. And I'm going to drive this point home with one more example. This is work that we did last summer, actually. We're working on a large project that's funded across the US to look at nutrition in vineyards. And one of our trials is trying to correct magnesium problems using foliar sprays. And what happened, of course, in this vineyard, it had a history of magnesium problems. So we knew we would get some magnesium problems. We went out shortly after veraison and collected samples. So I'm just going to show you some pictures here of the samples we collected. These are what I'm calling a zero sample or no symptoms. These are healthy leaves. And then these are leaves that were first showing the most mild symptoms. We also collected leaves that got a little bit worse. We call those number 2. Number 3, a little bit worse. And then 4, these were the worst looking leaves in this vineyard. Now we knew this was due to magnesium, but when we compare, again, leaf to petiole magnesium concentrations from tissue tests, you can see in this graph, if you look first at the red points, that's the petiole magnesium. Initially, yes, it goes down from the 0 non-symptomatic to our first lightly symptomatic leaves, but you can see the relationship is clearly not consistent. However, when you look at the leaf blade on the bottom in green, we have a nice relationship with the symptoms. So once again, leaf blades are a better choice based on the information that I've been gathering. Okay, one final area I'm going to talk about as far as tissue testing is this idea of using dormant season pruning wood to predict next year's nutrient demands or next year's nutrient status in the vineyard. I really like this idea because, one, we have a lot of time at this time of year. You're not busy doing 5,000 other things in the vineyard. So it's a time when collecting samples can be done in a good systematic way. Typically, we're cutting the pruning wood off, throwing it on the ground so we have easy access to samples, and we have a lot of material. So if you could get good relationships, it would also give you a lot more time to plan your fertilizer management for the upcoming season. So we looked at this question, and really it's 2 questions. And they are, do nutrient concentrations in the dormant canes respond to varying inputs like, for example, leaf blades and petioles do? And then the other question is, can those concentrations in the wintertime in dormant tissue be a good predictor of next season's nutrient status? So again, I used data from this pot and pot vineyard where we had varying nitrogen, phosphorus, and potassium supply that's been carefully controlled. So now just looking at nitrogen, our tissue response in terms of what was in the dormant stem is shown in this first plot. And you can see, let me explain one thing. In 2011 was the year before we began applying different rates of nitrogen. And then in 2012 and in the 4 years following that, we were applying these different rates of nitrogen to the plants. You can see in that first year in 2012, we got a pretty nice response in relation to nitrogen input, but then it kind of faded and got much smaller over time. Plus, we see things jumping around from year to year. And indeed, when you take that data and then predict what's going to happen the following season, by actually, well, I mean, we actually measured what was in those plants, of course. So this is using that dormant cane value for nitrogen to predict what's in the plant in terms of leaf nitrogen. It's not very good. Our R squared is, pretty weak. However, when we look at phosphorus, again, now this is the response in the dormant stem itself. And you can see now a very nice kind of response over time as we're applying different levels of P to these grapevines. We're getting a nice response in the tissue. And indeed, when you use that value to try to predict what's going to happen next growing season, you get a really nice relationship. So I was quite happy about that. And finally, looking at potassium, this is pretty similar. You can see how it is somewhat related to the phosphorus slide that I just showed. And you get a nice response over time that gets larger. However, when you use it to predict next year's potassium in the leaves, it's not quite as good. I mean, the R squared it's not bad. It's 0.63, you know, it's 2/3 of the variation. So somewhat good, sort of happy, but maybe it's more neutral. Anyway, we're doing a lot more nutrient testing as part of this large project that I'm working on called High Resolution Vineyard Nutrient Management. This is just a slide here kind of showing the team and the various people that are involved in this project. And we're trying to reevaluate nutrient testing as part of this large project across the United States. So we're going to be looking at these things in further detail, including the use of pruning wood to predict nutrient status in different kinds of grape commodities. Okay, what am I doing on time? Okay, so I'm going to move away from tissue testing now and talk about the time of nutrient uptake by grapevines. Obviously, knowing when grapevines acquire nutrients from the soil should give us a better tool set in terms of managing our nutrition programs for grapevines, so that we can time our applications when the grapevine wants it. So I'm going to talk about two studies here. The first one was done on 22-year-old own-rooted vines. These were in the field. I'll show you a picture here in a minute. And then I'm also going to talk about and compare it to another study we did after that one, where we again used this pot-and-pot system to look at nutrient uptake in young vines. The goal for, or the goals rather, for both of these projects were pretty much the same: determine when nutrients are taken up from soil, and also try to quantify how much nutrients the vines actually took up from soil. And then a third goal, of course, was to better understand how nutrients are being recycled through the plant. So this is a picture of the old vineyard that we worked in initially. Here we're, this is my crew that I had at that time, taking the plants apart, or taking the canopy apart. You can see there's not too many happy faces here. This was a lot of hard work. And of course, this is the easy part, taking the top of the plant apart. The hard part came when trying to look at roots, of course. And so we had done a couple different, we actually changed methods halfway through the study, just because the first year we killed ourselves. So then the second year we changed our root sampling. The good news was it didn't matter what we did, because our root data came out not affected by that change. However, this took us usually almost a whole day to get the roots from for a single vine. So this was quite an undertaking. And of course, that's the reason why when I went to the young, when I wanted to look at what's happening in young vines, we went to this pot-and-pot system, because then it was a very simple matter of dumping the pot out, and then we can wash the roots off really quickly. So this saved a huge amount of time, which is why we did it. So the data that we generated looks like this. And so I'm going to take a minute here to cover, to explain this slide to you. This is the nitrogen content in the whole vine in all its different parts. Starting with the yellow at the bottom, that's the smallest, or the fine roots. And then moving up, we go to small woody roots, large woody roots, the very kind of charcoal color is the trunk. Then we have the lay-down cane, the canes that grew up that year, or shoots. And then the really bright green little area in the middle there, that's the petiole end, then the leaf end, and then finally the fruit end on the top. So this is over 2 years. And if we look in the first year, as you move across the season, as we go from bud break to bloom to veraison to harvest, if you just simply follow along the top of this plot, that's nutrient uptake from soil. This is because what we've done is we've calculated the total content in each of the tissue types. And then if you look at the way this, the way the different compartments change, you can see quite clearly, we start off at the beginning of the year, and as we move into the middle of the season, the nitrogen in the roots and in the trunk drops dramatically, and that's partly supplying what's ending up in the leaves. So it's basically being remobilized and transported back up to the shoot. So much of the nitrogen in these old vines is being recycled. And you see a very similar thing the next year. Indeed, the second growing season, it actually was a little drier, and it really looks like we got a little bit more remobilization in that year that was drier. So this is the old vines, 22 years old. Now, if we compare that to the young vines, next slide, this is the same basic thing. We made one small change, which was we no longer separated the very, very finest roots from the small woody roots. We just called all the small roots and kept them together. The reason for that is because the amount of work was just too much, obviously. I mean, not obvious to you, but it was kind of a career killer for me. This project took up way too much effort and led to one publication. So when we did the Young Vine project, we said we've got to figure out some ways to save some time. But okay, the main thing you can see here though is a very similar kind of pattern. And there's a break between the 2 years here because in this experiment, both years the vines were 4 years old when we started. You know, so the vines we harvested in the second season were planted a year later as well, because we wanted to have it be very, very much the same age. But again, you see, even in young vines, there's some nutrient cycling for nitrogen coming from mainly the smaller roots. Now, if we look at phosphorus, now shifting back to the old vines, so this is the same plot like nitrogen, except now this is phosphorus. You can see there's not quite as much remobilization. As we start the season out, especially that first year, yeah, we drop a little, but then it doesn't really drop quite like nitrogen did. However, in the second year, it actually dropped lower. And again, that year was a drier year in that particular study. All right, so here's the same data for phosphorus now in the young vines. And really, we have almost no mobilization or reuse of reserves for phosphorus in young vines. This actually makes a lot of sense because we're dealing with mainly phosphorus deficient soils where we're growing grapevines. So basically, young vines cannot remobilize P because there's just not enough P available to them yet. And they're focused on just getting P from the soil. Now, this is now potassium, again, switching back to the old vines. You can see with potassium, it's kind of like phosphorus. There's a small amount of remobilization, not nearly as much as with nitrogen. But we see two other things that are kind of interesting with potassium, and one is you can see how much more potassium ends up in the petiole, that bright green zone in the very middle there. That's the petiole, And that's a tiny tissue, you know, weight-wise, biomass-wise. So potassium does accumulate in the petiole, which is why, of course, California was focused on using petiole when they were developing tissue standards. The other thing that I'd like to point out is looking at the fruit, the purple, you can see how big of a sink the fruit is for potassium. It's a huge sink for potassium. A lot of potassium moves to the fruit, and it continues to happen until you harvest. Again, here's now the young vines with potassium. It's a pretty similar story. You can see how that petiole really jumps out, and also how the fruit is such a large sink for potassium. The other thing this shows, of course, is we did get some remobilization of potassium in these young vines. Okay, so now to make a summary of this in terms of timing, which is basically, if we would just follow that very top line of all those plots, what I've done here is I've just taken the averages for both years, combined them, and then looked at the major growth stages of the plant: bud break to bloom, bloom to veraison, veraison to harvest, and then post-harvest. And asked when, or what was the percent of the total nutrient taken up in those different time windows. And you can see from this plot, from this table, I highlighted the bulk of the nitrogen was taken up by bloom. This is in the old vines. When we look at the next nutrient, phosphorus, it was about equally split between bud break and bloom, and then bloom to veraison in terms of when phosphorus was actually taken up from the soil. And then when we look at the other nutrients, clearly the main time of uptake is bloom to veraison. In these old vines from the field, the micronutrient data was unfortunately just way too variable, and we could not accurately quantify that. However, in the young vines, in our controlled system, we actually were able to quantify some of the micros. And that's the first time that's ever been done. Kind of the bummer thing about that, I guess, from a scientific point of view, is it's not super exciting because all the micros basically were taken up between bloom and veraison, you know, the bulk period of uptake. But again, focusing on nitrogen at the top of the table here, you can see even in young vines, most of the nitrogen was taken up by bloom. Phosphorus, not quite as clear of a equal split between those first two growth stages. But still, phosphorus was somewhat taken up early. So what this means is you can use this kind of data to kind of decide when you're going to fertigate. And so you would want to supply more of your nutrition depending on what your particular vineyard might need at different times, depending on when the vine wants to take those nutrients up. And so nitrogen, of course, we need to supply that early here where we live in our climatic conditions because the vines are taking it up early. If we waited until after bloom to start fertilizing with N, we probably would not be very efficient. However, if nitrogen was not limiting, that's a whole nother issue. So another thing I'd like to just summarize from this work, and ignore the light gray stuff, the canopy demand column there, but if we just look at the uptake of what was actually changed from the point of bud break to harvest in both the young vines and the old vines, the amount is really small. So for nitrogen, we're looking at 14 and 16 kilograms per hectare. Almost, maybe not every soil, but nearly every soil can supply that amount of nitrogen through natural mineralization almost every year. So these values are very low. like in the early work on grapevine nutrition, you know, people were applying somewhere in the order of 50 to 100 to 200 kilograms of hectare of nitrogen in their studies. Now, of course, not all that nitrogen gets taken up by the vine because it's not 100% efficient, but still, you don't need that much nitrogen. And of course, as I'll cover in the next section, I think it's a good idea not to have so much nitrogen. So the main focus of this slide is to talk about how low the demand for nitrogen is. You can see with the other nutrients, the demand for potassium is quite high, or the amount even taken up from soil. That's been supported by a whole lot of other studies. But still, these values give growers a handle on how much nitrogen or how much phosphorus or how much potassium you might want to add to soil when plants are deficient. So you know how much to add to correct. And these values are not that high. So to summarize all this, these studies, and there's been 5 other studies where people have done whole vine nutrient uptake, like the studies I just showed, the 2 studies that I've done. And one of those other studies, they only looked at nitrogen. And then I think another one, they only looked at potassium. So there's not a whole lot of data out there. But our data differed from a lot of these other studies because we had much lower quantities of nitrogen needed and much lower quantities of potassium needed by our grapevines. Partly that's because we have soils probably that have high organic matter content and can supply nitrogen. And Western Oregon typically crops vines very low. So, gosh, I can't do that conversion very well. But in comparison to most other grape growing regions, our crop loads are about half of what other people tend to have on the plant. So that's part of the reason why we have lower requirements. The other thing, of course, that was really different was we see nitrogen uptake occurring much earlier in the season than other studies. And I think that's largely, again, because of the region we're growing in. We're mainly dryland viticulture, and we have lots of soil moisture early in the season, and we dry out in the summer. Okay, the other thing that we found that was kind of interesting was that we used a lot more nitrogen and phosphorus from reserves already inside the plant. So one has to account for that. And then, of course, the older vines used more reserve, or they recycled more nutrients. Okay, so that ends the first 2 parts of my talk today. And I thought I would take a little break here and have some time for questions. I'm going to stop sharing for a bit here so I can see people. And if Noah would help me manage the chat box.
Yeah, we can start with reading the questions that people are already asked. And then if anyone else would want to ask verbally, we can.
Okay.
There was a question at the beginning of your lecture. I think it was the slide that you showed on the correlation between the leaves, leaf blade and leaf pet. No, it was before. One of the first one, someone asked what was the method used for the leaf test. I think it was really one of the first slides.
So
before you talked about the tissue analysis.
So the methods that we use for our tissue tests are materials dried and ground. And that's pretty standard. Like normally growers here basically just send that to a lab, right? So you either collect a leaf or you collect a petiole, not a, you collect many, but you know what I mean? Normally you send petioles or you send leaves to a lab. What the lab does is they dry it, they grind it, and then they analyze it. And so the analysis is the part that I think the person's asking about. And for nitrogen, we do what's called the Dumas method, where you burn it and then you measure the nitrogen gas. That's been the most common method used across the United States by testing labs at the present time. All the other nutrients then are measured by what's called ICP, or inductively coupled plasma. That's something we don't want to get into here, but you basically do the same thing where you grind it up, but you digest it with acid, you squirt it in this machine, and the machine will tell you all the different elements. So that's the test that we use. Hopefully that answered the question.
Yeah, hopefully. Okay, so the second question, I think maybe we can leave it to the end. It was a question about the differences between managing vine nutrition in cold or warm climates. I don't know if you want to address it now or at the end. I think it's a more.
Yeah, actually, let's leave that one to the end if we can.
Yeah, that's a big one. Someone asked if there's thresholds to the cane test, to the dormant canes or stems that you show.
Right, that's a good question. So the dormant cane data that I have, as far as I know, I'm the only person that has this data right now. So really, yeah, no, there's no thresholds, but I was just exploring to see, can we use this in the future? And so that's why I commented about the big project where more people are going to look at this. Hopefully we get a better answer. So right now, no thresholds, unfortunately. And I can see the next question. Does rootstock affect conclusions? I'm not sure what conclusions we're talking about here, but yes, I would say rootstock affects everything. It has a big effect on nutrition, of course, but whether or not it affects thresholds, going back to the prior question, is not always so clear, because what we're testing is the scion, we're not testing the rootstock. But we know different rootstocks have different capabilities. And probably the best nutrient that we know about is potassium. There's been a lot of work to look at the ability of different rootstocks to supply potassium. And so we have good information on that. I can't remember off the top of my head, but there's a whole series of work done in Australia. There's been some in the US and some in Europe. And certain rootstocks are known to be low potassium rootstocks, and certain rootstocks are high potassium rootstocks. And it does relate to parentage of the rootstock, but I can't recall off the top of my head. But yes, rootstocks do have a big influence. And the other big one is vigor, of course. Most of the work that's been done on rootstocks with vigor, we don't really know how well those rootstocks take up, for example, nitrogen, but I have a feeling a lot of it is a lot of the vigor effect is due to nitrogen. Okay, so
in your potted experiment, was that on rootstocks or on roots? On your Pinot Noir experiments that you show in the pots, were these grafted or on roots?
The picture I showed was grafted vines, and they were grafted on, it was Pinot Noir on 10114 MGT. The thing about the pot and pot system is the rootstock doesn't play such a huge role because we're controlling what the vine's seeing. So we still could get the response we wanted because we varied the amount of nutrients that were being applied. And I'm going to talk about that next, so you'll get a better idea of that. That experiment was actually done twice. The first time we used donor-rooted vines, and then the second time we used vines on rootstock. So yes, that picture was from a rootstock trial.
What is the pH? We are
I couldn't understand the next question.
I guess.
Okay. What is my opinion regarding SAP analysis? I think that's the next. Oh, no, wait, sorry, I skipped one.
No. You have the, what is the characteristics of your pH in the soils?
Yeah. So we're acid soils here in Western Oregon. We have high rainfall in the wintertime, and it has washed all the cations down. And so we have low pH. A lot of our native soils are probably around 5. We recommend typically to bring the pH up to at least 6. But yeah, we tend to be acid, but I'm going to show you some work later in the talk because I've compared our soils here on the western side where it's wet and humid and acid to soils on the eastern side where it's arid, dry, and basic. And so I can get into that a little bit. I don't understand the next question. Sorry, I don't get that one. But the next one I see is, what's my opinion regarding SAP analysis? I guess that's kind of a tough question for me to answer. It has its utility, yes. I think one of the issues we have to think about with SAP is the time of day you collect SAP is extremely important, and nutrients fluctuate through the day. It's one of the downfalls of using SAP. I don't know, we'll see. There's hopefully some SAP analysis going on in this big project that I mentioned. I'm not actually doing any myself. I'm not a big believer in it. So my opinion is really not too high, but that's my opinion. You know, I recognize some other people might really like that idea. But the problem in my view has been this issue with fluctuation through the day, and time of day influences your samples a lot. Okay. The next question is regarding soil test relationship between soil K, pH, tissue sampling.
That is not a question. That's someone who has comments or maybe questions. Ran, do you want to unmute yourself and ask your questions?
Mm-hmm. Can you hear me? Paul, no? Can you hear me? No?
Yeah, we can hear you.
Okay. Let me just briefly introduce myself. I started working on vine last year with the main winery in Israel, and we are questioning everything, okay? And we start with a very wide survey of over 300 vines across Israel. And the first thing I see is that there is pretty solid correlation between the soil test and the leaf analysis. And that is regarding Olsen B, which is what we are using here in our calcareous soils, and calcium extraction with either, potassium extraction with calcium chloride, it doesn't matter. And it seems, again, from very brief observation of only one season, that it does respond. I mean, high availability of these nutrients, not necessarily nitrogen, and the leaf analysis, we tested only in the spring at flowering. It correlates pretty solid. I don't remember, but it was fair. Mm-hmm.
Noah, could you help me a little bit, because coming all the way to me,
I must say, I
a very small amount of that.
Ran, I think your connection wasn't really well. Did someone here run good and can repeat it, or we'll try again later? Anyone?
I'm sorry.
I couldn't hear it as well. I think it's connection.
Okay, sorry. We can chat later.
Yeah, we can chat later.
Nobody heard him, Noah.
Nobody heard
Nobody
it.
heard
Okay.
him.
Okay. So Ran, I guess your connection is not as well.
Sorry about that.
Yeah.
Let me see what time it is. I would like to move on if that's okay, unless we have another burning question. Let me see. I can answer a couple of these very quick. Are the vines irrigated? The vines in my pot and pot study, of course, are irrigated. Most vines are now irrigated, but there's still a lot of vines in Western Oregon that are dry. They're not, no irrigation is used. But we get 1,100 to 1,500 millimeters of rain. So about 2 to 3 times what the wet areas of Israel are getting. Let's see. The timing of nutrients uptakes was understood, but when to apply, when to fertigate. Well, I would say it generally agrees with when uptake is occurring, but it also depends on your situation. If you fertigate, you're making nutrients available immediately. And if you're irrigating, pretty much the roots are under the drip emitters. So you have a pretty rapid ability to affect uptake. If you're broadcasting fertilizer and not irrigating, then you've got to think in advance. Now you've got to put that fertilizer down much earlier. So it depends on whether you're fertigating or you're broadcasting and how much irrigation you're using. Okay. The last question, what do I mean? Uptake post-harvest, that means after the fruit is mature, was there any nutrient uptake between that time period and pruning? And for the most part, it was pretty small. Okay, I'm gonna move on and share my screen again, if that's okay. Yeah. Before I do, can I ask, am I clear to everybody? Can you hear me well? Okay.
Yeah.
All right, so moving on. Again, it's not acting right. Okay, there we go. So part 3 of this talk, and let's see, we're about an hour in. So I'll try to get through this part a little bit quickly because I think the last part may be of more interest. So I'm going to talk now specifically about these pot and pot experiments where we looked at N, P, and K requirements for Pinot Noir. I think this will be of interest because we were looking at impacts on quality. And so I explained earlier, we did this twice. We did 2 studies in this pot and pot system where we manipulated N, nitrogen, P, phosphorus, K, potassium. The first time we did own-rooted vines. I'm not going to talk about that project because the second project, we had much more comprehensive data. In both of these projects, however, we used this pot and pot system, which was a huge investment for my lab to build. I want to point out here, the second project, the MPK2, which I'm going to talk about, we had a lot of help, and it's because we looked at a lot of different things. So we had viticulturists and enologists helping on this project. And so all the little symbols there with the little beaver head, those are my colleagues here at Oregon State University, and the other folks are USDA folks. And in this experiment, I'm not going to go into all the detail because it would take all of our time up, but we tried to look at everything we could think about to really get at understanding the physiology of nitrogen, phosphorus, and potassium supply in Pinot Noir. And one of the things we did, in addition to having the system that's highly controlled, is we wanted to also account for indirect effects of the nutrients. And so the key thing, we knew the nitrogen was going to affect vine size or vigor, right? How big the canopy grows. And so we wanted to control for cluster shading. So we did that by measuring fruit zone PAR measures, you know, sunlight in the fruit zone. And then we pulled leaves in treatments to match so that we could get everybody to match up with solar radiation. However, this was only successful to a point. Our two lowest nitrogen treatments, which I'll show later, we could not do that because they were just too weak. And if we would have tried to pull leaves off the other treatments to match those low nitrogen vines, it would have basically destroyed the experiment. The other thing we did was we measured vine water status. Mainly, we were looking at soil moisture. We did this daily by TDR, and then we correlated that with leaf water potential values. So then we could manage things very carefully so that we applied different amounts of water to different treatments when the vines were affected by, you know, had a growth effect basically. And we're using more or less water. So the bottom line is my technician pretty much was here every day. This experiment was a lot of work, and it also took years to build up because we had to grow the vines until they were mature enough to carry fruit. So it was a big investment. And why would I go to all this trouble? Well, basically, it's a simple thing. It was to control vine access to nutrients mainly and water. And the reason is because we had already done trials in a lot of our vineyards here. And two things happen. The inherent soil fertility sometimes overrides what you're trying to do. But the other big thing, of course, is that spatial variation that's out there in both fertility and depth of the soil profile a lot of times overrides your treatment effects. So it's hard to get highly controlled data. And so hopefully you'll find this humorous. When I saw this little cartoon, I had to laugh because, you know, to me, here I am out there carefully metering out these little bits of nutrients. And the biodynamic guys just have no respect for that. All right, so this is what the plots look like. I was actually standing on top of my truck so I could take these pictures. This is, and I'm going to focus first on nitrogen from this trial, but you can see pretty clearly here, I'll circle it, the low end vines really stand out. So each one of those 5 vine panels are one of our lower end treatments. And so I'm going to show a few plots like this. And basically, this first set of plots on this slide, what I'm showing is bloom leaf nitrogen at the top, or leaf blade nitrogen, and then veraison leaf blade nitrogen on the bottom slide. And the black dot, or the black symbol, is our highest nitrogen. And then as we go down, it goes from, you know, kind of darker green to more yellowy green, kind of the way the vines do. So that kind of made sense to me to color it that way. You'll notice there's this darkly dotted line where the yellow pops back up. That's because I decided, well, after that 3rd year, let's give that lowest end the high end rate and see what happens. So that's what that data point is. But you can see from this data how our nitrogen treatments in different rates were having the intended effect. So we were varying nutrient supply as we wanted to do. And you can see there's a nice gradation of vine nitrogen status with our inputs. So what was the impact of that? So here's now vegetative on the top, reproductive on the bottom. So it's prune weights on the top, and then yield on the bottom. I do want to point out that these vines were thinned to 1 cluster per shoot, because that's the standard practice here. So they're not carrying the maximum that they could carry. But we needed to do an experiment that was similar to our production systems. The important thing that this shows is how much more prune weights are affected by nitrogen status than is yield. You can see that even our second highest nitrogen rate was really differentiating itself from the control. And then we were really hurting plant growth in our 2 lowest nitrogen rates. And indeed, if you focus on that, the very middle nitrogen level, you can see on the top graph, we cut growth in half practically. But while we did that, we had no impact on yield. Yield was not significantly different on the lower plot in that treatment. So that's a really nice target for the nitrogen level that you ought to have. And I should explain also, we tend to have a vigor problem in Western Oregon. Our vines tend to be too vigorous. So there's a desire to get some control. So hopefully that all makes sense. Now, what about impacts on the must? Well, it's juice bricks on the top, really nothing happening there that's consistent. Juice yan on the bottom. Excuse me, I've got a bug in my office. Juice yan on the bottom, you can see a nice gradation, just like we had seen with leaf and petiole, or leaf. I only showed leaf. Of course, we measured petiole here too. But anyway, the point is we get a nice gradation in yan, and many winemakers would be very upset at a lot of these yan values. Of course, some of them are clearly too low, but what's interesting is we made wine from these trials. These were not large plots, so we had to do small lot wine. We did them in these 3 kilogram small containers with submerged cap. And here is the impact of must yan on fermentation rate. So you can see these are all the individual replicates from all the different treatments with varying nitrogen. The main thing is we really don't see a large change in fermentation rate until we start to get near 100 milligrams nitrogen per liter. So that's kind of what I would see as a nice recommended level for yan, instead of 200 or 250 or 300 or whatever. The industry has different values that they're using, but almost everybody's using pretty high values. And the main argument is because they don't want to have stuck fermentations. Well, we're not really having a problem here all the way to 100. In fact, we get well below 100 and we still don't really have a problem. Yes, as we got, our very lowest yans were very low here. We were in the ballpark of 25 in the last year of the study in 2014. Those were taking almost 20 days to ferment. So yeah, that's a problem. But they still completed fermentation, which is a little surprising to me. Here's just a picture of wine color, just to give you an example of what was happening as far as this particular parameter. And so these are just the 5N rates, an example of our wines. In this year, by the time we got to this year, those ones that are highlighted here had the same sunlight exposure on the fruit. And you can see we did start to get some darkening or better color in the middle nitrogen rate, even though cluster exposure was the same. But of course, you can see the advantage of having really low nitrogen in terms of color on the very end. Really, really good color in our lowest nitrogen. But of course, our yield there is about half of what the control was. So we also looked at wine aroma. This is with a partner at Oregon State University. I'm not going to show all this data because, oh man, it's a lot of data and it's like it gets complicated fast. So I'm going to summarize it. Basically, nitrogen had a really large impact on wine volatiles. And surprisingly, to some degree, it affected the yeast-derived volatiles, or what we know as the fermentation bouquet. The terpenes and the norisoprenoids, which are in the berry, which are important aroma volatiles, were not changed nearly as much as the yeast-derived esters and other compounds that come through yeast metabolism. The other thing is our low nitrogen wines had lower sulfur compounds, like the nasty kind of ones, that give you that rotten egg sort of aroma. This is completely opposite of the literature, and in fact, it's the argument that most winemakers use to want higher YANs. Well, we saw the complete opposite in this study. And maybe that's because we use a submerged cap. That's a question I can't fully answer. We also did look at some sensory analysis of this. We only looked at 3 N rates, and I'm going to show 1 year data here, but no surprise. The high nitrogen wines were more herbaceous, had kind of green bean notes sometimes, but also some red fruit and spice. And then medium N were more kind of like the desired kind of things that we think about for Pinot Noir aromas, raspberry and dark fruits. And then finally, the lowest nitrogen were more floral aromas. So all in all, the lower nitrogen fruit had a more pleasing aroma. So what I've done here is kind of summarize this data in a way that puts it all in one place based on Veracean leaf nitrogen. And so what I've got here is sort of like a thermometer, if you will, with going from basically low to moderate nitrogen in the leaf blade. And so if we're around, in our system with Pinot Noir, 2% leaf nitrogen at Veracean, that's going to give you a must-YAN of about 140 milligrams of nitrogen per liter, which is one of the common ones that a lot of people use. At that point, we're having no impact on plant growth. If we drop down a little bit lower, about 1.85, that's where we see must-YAN coming in around 100, which is, I think, a better target value for us to use, because we have these other positive benefits with lower nitrogen fruit. When we get to a value as low as 1.7, now we're consistently reducing yield. And of course, we were reducing prune weights at a slightly higher nitrogen level too. We did not see effect on some of the other physiological parameters until we got much lower. So fruitfulness, the number of clusters per shoot, fruit set, photosynthesis. We don't see any effects on those until we're all the way down to 1.6% nitrogen of Veracean. And a lot of those weren't even consistent. And then I just, as a final thing, if you ever get to 1.5, you only have to be there in 1 year and you're going to have a yield reduction. So that's kind of our measuring stick or threshold values for our production systems that apply to Pinot Noir. So that was nitrogen. I'm going to cruise really fast through phosphorus and potassium actually, but I'm going to cover that quickly. Here is phosphorus. These are our vines that were under the lowest level of phosphorus for 4 years, and they were getting no P for 4 years. They look pretty good. But similar to the nitrogen story, this is what we wanted to do. This is our vine phosphorus status based on leaf blades. Bloom on the top, Veracean on the bottom. The take-home story here is we were doing what we wanted to do. We're getting different levels of phosphorus in the plants. Our impacts were really small. We did not affect either vegetative or reproductive growth until we were in 3 years with no added P. And it wasn't a huge drought. So Pinot Noir is pretty damn efficient at using phosphorus. All right, I'm going to go through some of this a little bit quicker now. We saw no effect on flowers or fruit set with P, which is completely opposite of the literature. Again, I think it indicates that Pinot Noir is particularly efficient with phosphorus. We did see red leaves by the end of the growing season in our no P vines. And it was really interesting how dramatic it was, because right around harvest, the leaves would just turn red. And we didn't get much indication prior to that. Okay, I'm going to move on. Basically, we got phosphorus. Now these are the means. Sorry, I didn't plot the raw data like the nitrogen, but this is again fermentation versus juice phosphorus level. No impact whatsoever. We got down to pretty low, 25 milligrams P per liter. No impact on fermentation. So impact on aroma volatiles were almost non-existent. And we never saw the same thing happen in more than 1 year. So I can pretty much rule those out. So the bottom line with P in this system is that it's a viticulture issue. The only thing we affected were growth and yield. Those were the most sensitive things. And I gave our growers values that correspond to that, or threshold values. Now, would these apply to other systems? Not exactly, but they'll be close. So because of time, which I was worried about, and I imagine some of you are starting to get bored of hearing my voice, I'm going to go a little faster here. So potassium, again, we manipulated potassium similar to nitrogen and phosphorus. Now these vines, I don't know if you can see that picture as clearly as I can, but they're starting to look a little ugly. These are no K vines. After 4 years, they were getting no potassium. I'll show some better pictures here in a minute. Bottom line, this is like the other story. We did what we wanted to do, right? This is bloom leaf K, veraison leaf K at different levels of K supply, and we're having the gradation that we wanted to see. Again, we look at growth and reproduction, prune weights on the top, yield on the bottom. Similar to phosphorus, really. We did not see a big effect until the very end. And then yield really dropped, and prune weights. And you'll see why. This is the key thing. The most sensitive thing affected by potassium supply was juice pH. We saw an impact on juice pH before any other thing we measured. So juice pH is a thing you need to keep an eye on when you're managing potassium. Okay, here's some ugly pictures. This is like the first obvious leaf symptoms. So this was in about mid-season. We start to see symptoms in our, in this case, this is the no K vines. The other thing that happened is we caused bunch stem necrosis to occur. I'm not going to go into that any further detail other than that's another symptom. But here's a really good picture to show you how bad these vines look by the end. This is our no K vines in the last year, and you can see why yield and prune weights were so low. Obviously, we went way beyond deficient. We're, basically nasty. I don't know how else to say it. And this occurred when leaf K was at 0.4%. Now, if we look at fermentation, interestingly enough, again, even though we had a pretty good range for potassium, it had no impact on fermentation rate. Aroma, similar story to P. There were a few effects that were there, but honestly, for me, they weren't enough to really highlight anything, especially because what you're going to be affecting more importantly is juice pH and wine pH. So I came up with this similar kind of thing with nitrogen, where we have kind of a thermometer, with, if you will, of veraison K status and the various things that happened. The bottom line is we're proposing a critical value here of 0.6%. in leaf blades efferation, that actually agrees with the really old French standards, which is interesting. Once you get below 0.6 and you get near 0.5, that's when we start to see must pH being too low. And that's also when we start to see the very first symptoms of late bunch stem necrosis. We don't affect growth really until we're much lower. I will point out this is Pinot Noir at low crop load. And so I don't know how good these apply to other systems. Okay, let's see what time it is. All right, that took about 25 minutes. I have one more part to cover, which is going to be on mycorrhizal fungi. So I can stop for a minute here, take some questions, and actually maybe everybody could take a break for a few minutes, make a cup of coffee,
Sure.
whatever you guys want to do. Go to the bathroom maybe, and I'll try to answer questions.
No one wrote questions. Anyone has a question for Paul at this point?
Yes, can I? At the beginning of your, can you hear me right?
Yes.
Okay, perfect. At the beginning of your, I have two questions. At the beginning of your presentation, I saw some of the values that were significant were calcium and Ca in the very beginning. And do you apply calcium to the vines, and how you do it if you do?
Okay.
What size? Mm-hmm.
So no one fertilizes typically with calcium, although we lime the soil, right? And so that's your source of calcium typically, right? So, and that's more about a pH issue, I would say, as much as it is about calcium. It's really, really rare to find calcium deficiency. And I have never seen that here in the field, even though we have acid soils. I have never seen it in any of our vineyards. And as I'll talk about in a few minutes, in arid environments, calcium is probably going to be too abundant, if anything, right? And start to cause problems with iron or even phosphorus. So that the calcium issue is really more about managing pH, I would say, unless you have an extreme situation. And I have seen calcium deficiency, but the only time I saw it was when someone was doing it in a hydroponic system. In the real world, I've not seen it. Have you had issues with calcium that you think are deficient?
What I heard and seen is that calcium, when applied by foliar applications, might help with mildew or fruit disease. That's the only thing. That's what I wanted to know, if you did apply in such a way.
Okay. That's not something that's commonly done here. But how do I answer that right? There's a lot of people who spray a lot of different things that they think gives them a benefit, whether it's for disease control or nutrition. And rarely is there scientific data to back it up. And you know, science isn't perfect, but as far as calcium goes, in terms of disease suppression, it might play a role. I'm not going to say it doesn't, but it's not a common thing to do here. And so I don't really have a good answer for you. I'm sorry. What was your second question?
Because I hardly need to irrigate my vines. I tend to follow foliar applications, and I wanted to know if you have any idea, if you think it's this one or the other better, foliar or root application through the irrigation.
I think personally, my opinion is root is better, but it depends on the exact situation. So for example, if you have plenty of nitrogen and you have plenty of vigor, then you probably don't want to apply nitrogen to the soil. But sometimes people have enough vigor, but they still have low yan. And winemakers want them to have higher yan. And so in that case, I would say foliar is a good way to go with nitrogen. With other nutrients, it's not as effective. And the reason primarily is because we can apply nitrogen as urea, which is uncharged. And because it's uncharged, it crosses the cuticle well. Whereas many other ions that we apply as a fertilizer for foliar have a hard time actually getting into the plant. So I think you can tweak things a little bit with foliar, yes, but a primary approach would be, in my view, would be soil for most nutrients. However, the one project I'm working on right now, where there's magnesium deficiency, we cannot seem to correct it with soil applied, but the growers have also been a little nervous about applying really high dose. And so in some cases you have to apply a higher dose. So I do think there is potential for foliar in many different things, but for the most part, personally, I would rather see it come up through the root system.
Thank you.
You're welcome.
Paul, there's 3 questions on the chat if you want to address them, or if you need a break, we can address them at the end. So your choice.
Okay. Let's see. How is everybody doing? Like, you're okay? You're not bored? You're not?
You're not bored.
Okay. You know, I don't know. I'm getting kind of tired, so I'm not sure what I'm saying at this point. So hopefully, nutrition balance in terms of pests and disease. Yeah, and this is all about nitrogen, really. That's another reason to not give so much nitrogen to grapevines, so you don't have such an obvious, lovely food source. However, certain pests are attracted to low nitrogen status. And so, it's not always a clear answer. The advantage of low nitrogen, however, in terms of the big problem that we have, which is powdery mildew, again, you get an advantage with low nitrogen because you're going to get more airflow. So you're going to reduce powdery mildew. That's our biggest problem. I don't know if it's as big a problem in Israel. Have I investigated correlation between elements and phenols? What is the best method for measuring phenols? So that's a hard question. I have looked at it. It was in one of our papers, and we did find that phenolics in the fruit are increased when you have lower nitrogen. And that happened independent of light exposure differences. So yes, we have seen that with nitrogen, but not with phosphorus or potassium. As far as methods for measuring phenols, I think, the standard is follincalcital, which is a reagent you buy. It's kind of toxic stuff. That's how you would measure it in a more simple way using a spectrophotometer. The other way would be to go HPLC, but not too many people have that instrument laying around in the winery. Okay, the problem with calcium is how to reduce the effects of calcium. That's a good one. I had a feeling that might come up, actually. Reducing excess or having too much calcium is not an easy thing to correct. You can try to add both potassium and magnesium because they're the 2 other cations are going to compete for calcium uptake, but still it's really, really hard, and especially in arid regions with calcareous soil. I mean, there's just so much calcium, it's really hard. But I would think it's also typically in those situations, you're drip irrigating. And so over time, you keep drip irrigating, you can start to get rid or leach some of that calcium away. So I think when vines are younger, maybe you're gonna have more problems, but over time, by adding some potassium and also irrigating, you can maybe get rid of some of the high calcium problems. But that is a tough one to solve, and I don't have a good answer for that. I'm sorry. And it's kind of outside of my wheelhouse because we're in the opposite world mostly with acid soils. Yeah, this is a great question. Should you spray potassium to increase sugar? Yeah, you could. I've tried that twice and saw no effect, but I know some other people who have done it and seen effects. So if it works for you, fine. Yeah, I don't have a problem with that. I mean, I don't think there's really a huge environmental concern with spraying a little bit of potassium on the foliage. But honestly, like in our low potassium vines that we were really stressed, we had no impact on sugars in the fruit. So I'm not a big believer in it, but I know there's many people who have published on it and some growers swear by it. So, you know, if it's working, great. Let's see, what about working with compost tea? That's a huge area to cover. I think especially applying compost to the soil is great. In fact, going back to the calcium question, that's one way you could maybe help alleviate some calcium issues, although it depends on the compost, because some compost sources have really high calcium. So you got to watch out for that and understand what you're applying. I'm not a believer in compost tea per se. A lot of people use it. A lot of people make a lot of claims. I see no scientific evidence that shows that it does anything. And I have a colleague here in my unit who worked on that. It's a long time ago, but they saw nothing. Now, part of the issue is control. When you're talking about compost sprays, a lot of times the people were claiming they saw something never even had a control that they sprayed water. And so what they were doing, they were getting some disease suppression, but it was because they were spraying, it was the water, not the compost tea, you know, so they reduced some powdery mildew with water. All right, I'm gonna move on and go to the last section. I'm glad everyone's still there and you're not bored with me. So we're gonna talk now about mycorrhizal fungi. This is something a little bit, I would say, near and dear to my heart because this is what I started working on for my PhD. And I'm going to go a little bit fast. So, but I'm going to provide PDF of all this. And then if you guys have questions, just email me. I'm happy to answer questions. So first of all, what's a mycorrhizae? A mycorrhizae is literally fungus root. We've known about this for a heck of a long time. It was discovered in 1885. That's damned amazing to me. And recognized as a symbiosis only a few years later. I mean, that's really phenomenal. I'm showing two pictures here. One is of an ectomycorrhizae. Ignore that. Grapevines don't form ecto. Grapevines form mycorrhizas with arbuscular mycorrhizal fungi. The picture on the right. And these fungi are very different than the ectos. And so there is confusion out there about that. So I wanted to make that very clear. So to point out, there are different kinds of mycorrhizae. Here's all the different kinds in a large classification. The ones you're worried about as grape growers are the top ones. They're the most common, they're the most ancient. Almost all of our crop plants form this type of mycorrhizae if they form a mycorrhizae. And they're incredible. So this is where mycorrhizae are cool. The picture on the right is a present day arbuscule. This is a tiny little tree structure inside an individual root cortical cell. This is where all the action is. So the fungus makes all this surface area and the plant allows all that to happen. It allows that fungus to grow inside and make all the surface contact. And that's where things are being exchanged. Carbon is going from the plant to the fungus. Nutrients are coming from the fungus to the plant. And the most important one is phosphorus. The beauty of this is it's been around for an incredible long time. And you can see the picture on the left. It's not quite as pretty, but you know, it's 400 million years old. And one other little slide here, which is kind of fun, using a molecular clock approach, the group of fungi, the Glomeromycota, are the arbuscular mycorrhizal fungi. They evolved probably 900 million years ago. It's an incredible amount of time. And so there's been an exceptional period of coevolution between the plant and the fungus. And just to put it in perspective, that's when dinosaurs were around and when Vitus evolved. So these guys have been there way longer. Why are they important? The main thing is they enhance nutrient uptake, and phosphorus is the big one. Other ones that are most common are potassium, zinc, and copper. Now there's a lot of papers published on nitrogen and showing that mycorrhizae help with nitrogen, the arbuscular mycorrhizae, sorry. And it does in some cases. I actually had a grad student finish with me recently. We could find no evidence of nitrogen uptake by mycorrhizae in grapevines, but we're still looking. The other thing they do is they enhance drought tolerance of plants, and they help suppress root diseases. So number 2 and 3, enhancing drought tolerance and helping with disease suppression, is sometimes related to number 1, which is improved phosphorus nutrition of the plant, but not always. The last thing they do is they help stabilize soil aggregates, which is really important in certain systems where erosion is a big deal. And of course, the reason they do this is because the fungus inside the plant, so here's a picture of a root of grape. The brown is the very outer cell layer that was not perfectly cleared. All that blue stuff that's being squished out is the mycorrhizal fungus inside the grape root. But the reason that they can do this is because they're inside the root itself, but then they have this external phase. So this is the hyphae now that are outside the root. And what I've done here is in a very sandy system is just gently tease this apart so you could see the hyphae connecting soil particles. And so these hyphae are very tiny and they explore the very tiny niches of soil. This is why they help plants so well. So a big component of them is that being the right size for the job is important. Here's a few more pictures of mycorrhizal colonization of grape roots. Hopefully those are clear to everyone. Basically, all the blue is mycorrhizae. And more critically, this is arbuscules in grape roots. This is where all the action happens. So this is where the plant and the fungus are exchanging carbon for minerals. And it's amazing, from my experience, how many arbuscules are in grapevine roots. So I consider grape a super host for mycorrhizal fungi. Now, as I've already stated, they're most critical in low P soils. This is a soil map of the US. The important thing here is there's, in the Willamette Valley on the west coast over there in Oregon, there's like the yellow thing in the middle and then the brown around it. The brown around it is altisols. That's where all of our vineyards are. Altisols are low P soils. And an example of how mycorrhizae affect plant growth in terms of grapevines, in a mollisol versus an altisol, so a high P soil versus a low P soil, you can see the plant doesn't actually need the mycorrhizae in the high P soil in terms of growth. But in the altisol in the red, the plant just stopped growing when it didn't have mycorrhizae. Now, in order to do this experiment, I had to sterilize the soil so we could test the idea. Now, if you look at what happened, it's very clear that this is shoot phosphorus in those different treatments. The plants in the red bar, or the red soils, which was the Altisol, they were phosphorus deficient. When they're mycorrhizal, then they have enough P. The other thing that happened was we saw potassium increase in both soils due to mycorrhizae. And I just wanted to point that out because I've seen this consistently. This is another way to display a lot of data, which I don't have time to explain, except this is an experiment where we were comparing 4 different mycorrhizal fungal species and how they affect grapevine growth. All 4 of them on this graph stimulated growth a lot. It was, vines were 200% bigger than the non-mycorrhizal plants. And then on the other axis, we can see that phosphorus was the nutrient that was affected the most. That's clearly a good thing. And then we also see potassium being a positive effect of mycorrhizae as well. Whether or not that's a good thing depends on whether or not you have the right pH in your winery. The other thing to point out here is the very bottom points here are the green, that's nitrogen. They're all lower than the non-mycorrhizal plants. So I mentioned that earlier. So far, we're not seeing an impact on nitrogen. Okay, so looking at your soils in Israel that are being used for viticulture, there were 2 really common ones that I found, the Terra Rossa soils and the Renzina soils. And I think the Renzina ones are actually split again into the pale ones and the brown ones, if I recall. Either way, these are very low P soils. So mycorrhizae is probably pretty important. And indeed, this is a paper, and I know this group was doing some other work. Basically, on these 2 different soil types, plant production, plant growth is severely restricted by phosphorus. Now, this is not grapevine work, but other work. Some other vineyard soils that I could find are the brown forest soils, which I don't know exactly what group they are in terms of our US system. And then another common one in vineyards are vertisols, which are these shrink swell ones, the ones that really develop big cracks. Now, both of those are kind of variable in terms of P. Some are very low, some are more moderate. But again, some of them are low, so mycorrhizae is going to be playing an important role. Next main thing about mycorrhizae is that they're clearly more critical in drier conditions. And I'm kind of running out of time, so I'm going to skip a little faster here, but in my point of view, this is mainly northern. I cut off the Negev desert on the bottom here, but this is just a Google Earth shot. That's where you guys are growing grapes. This is where we're growing grapes. It's really different. This is the Willamette Valley right here. And then all around the edge of that is where our vineyards are. And I mean, look how green this is compared to where you guys are. Clearly, you guys are growing under arid conditions, even in probably some of your wetter areas. I know there's probably a couple spots really high up in the very northern part of the country that get maybe close to the amount of rain we get, but I don't think it's quite the same. And of course, we do grow some grapes on our arid side of the state, but mainly that actually occurs north of us in the state of Washington. And in fact, I looked at grapes there, and this is a study where we were looking at how much irrigation affected mycorrhizal fungi. This is just an example of why mycorrhizae are important in more arid conditions. This was the experiment. 3 treatments is pretty simple. Standard irrigation, which is full vine ET at 70%. So you estimate how much vine would use if it was allowed to use as much as it wanted, and you apply 70%. That's kind of the standard practice there. It's called regulated deficit irrigation. So this study, they applied an even greater deficit early in the season during the first stages of berry growth, or they applied a greater deficit later in the season where they dropped that to 50%. So this experiment was just a nice opportunity to look at this question. here's what those plots look like. Looking down the rows, on the top is the control. You can see it's a little bit more green growing into the alleyway. And as we go to both of the deficit treatments below, we are limiting growth a little bit, but looking down the main row, you can still see the vines they're pretty happy. But when we looked at mycorrhizal fungi colonization, it was a little bit striking. It didn't matter when the deficit was applied, both the deficit treatments were much more reliant on mycorrhizal fungi. So we saw arbuscles in the roots go up significantly. And I should point out that we came at this after this was already applied for 2 years in the 3 years that we looked at it. And so we didn't catch this right at the very beginning, but clearly it shows that mycorrhizae are more important when it's drier. So there's no doubt in my mind that mycorrhizae are important in viticulture in Israel. And so these pretty little guys are your kind of hidden friends in the soil. However, most likely they are already working very hard for you. Maybe not in every case, but all right, I'm going pretty good. I'm going to cover this pretty quick, but basically, even though mycorrhizae are probably doing the job that they should be doing for you, you still want to understand how to manage them so that you can derive the most benefit. And so really you have two options here. You can enhance what you've got in the soil by choosing agricultural practices that are not going to hurt them or that will favor them, right? I want to talk about some of those. The other option is if they're absent or very, very low population in the place where you're at, you could inoculate with fungi that someone else hands you or grows for you. And this is the way we grow our cultures of mycorrhizae. We use a 2-plant system, sorghum and clover, in these little trays. Basically, you have to grow arbuscular mycorrhizal fungi on a plant. That's the only way you can produce them. We always choose this because they're plants we don't typically have in our systems, and we don't want to carry any pests to the grape. But when do you want to enhance, or how do you enhance the population that you have? The most important thing is to avoid really long fallow periods. So periods where there's no plants on the landscape. In other words, you need to maintain host plants in the system. And this is really more important probably pre-plant when you're planting a vineyard, because the mycorrhizal fungi have lost the ability to grow on their own. Part of that whole thing about long evolution. And indeed, here's a really good example from Australia. It's a completely different system, but this is where this whole idea of long fallow disorder came from. So they were having problems when they would do these long fallows. They were trying to save water, so not crop every year. But even when they would add peat, the plants wouldn't grow. Then they figured out, oh, if we add mycorrhizae back, now the plants grow. So this is where this whole long fallow idea came from. And so you want to avoid that. But cover crops are the tool that we have as agricultural people. Of course, they provide lots of other benefits, which I'm not going to go into. But in my mind, they are the most important tool you have to manage mycorrhizal fungi. But you have to avoid non-host plants. So there are a few plants to avoid. And I would say pre-plant, this is what you want to do. And you would want to put them, over the whole landscape. Okay, the other things to do in terms of managing what you have is avoid harmful chemicals, soil fumigants, soil fungicides, and readily soluble fertilizers, phosphorus and nitrogen in particular, and then also reduce tillage, because that destroys the mycorrhizal network that's in the soil. And then the plant has to pay extra carbon to rebuild the network. Okay, so this side kind of summarizes all the bad stuff to avoid. Fumigants, soil fungicides, readily soluble fertilizers, and tillage. And I'm going to go through this very quickly now. This is just data from this big survey I did of vineyards in Oregon for mycorrhizae. One of the things we found was when you used foliar phosphorus, you reduced mycorrhizal colonization. And it affected arbuscular colonization much more than just the presence of the fungus in the root. This is an example of where using phosphorus is going to have a negative impact on mycorrhizal fungi. Now this is data from the eastern side where it's dry. A similar study we did where we did a survey of mycorrhizal fungi and colonization. Here on the dry side, we found that actually soil nitrate was more highly correlated to mycorrhizal fungi than any other nutrient in the soil. So high P and high N can both have negative impacts on mycorrhizal fungi. Tillage is well known. There's been tons of studies. I just mentioned them here. And we did, one of our studies, we found the same thing. In this case, this vineyards were using in-row cultivation. And so this is a comparison where they were using in-row cultivation to control weeds versus herbicide. And when you do cultivate in the vine row, you knock down colonization by mycorrhizal fungi. Okay, now moving on to the other thing to do, which is inoculating vineyards. When's this needed? Basically, I already covered all this stuff, but it's when you treat your soil like dirt, or you're really bad to it. Fumigants, long fallow periods, or following a history of non-host plants. I gave an example here because in Oregon, hazelnuts are a big crop, but they're an ectomycorrhizal host, and they also keep the entire orchard floor free of all other plants, you know, so they can pick up the nuts. So in that case, you lose the arbuscular mycorrhizal fungi. I don't know of a particular example in Israel that would be the same. All right, I've done a bunch of inoculation trials when I first started. I'm not going to go into this other than to say it only worked once out of 5 different studies. And it was because both soil phosphorus was low and the fungi there were low. In most cases, your native population is probably going to be just fine. Now, I'll end here and I'll just put all these up. There's a lot of people selling mycorrhizal inoculum, and I even found some in Israel. They're everywhere. It's a really tricky thing, and you have to be really careful about buying mycorrhizal inoculum. It's expensive to produce. There's no standards of testing for any of this stuff, and so you really don't know what you're getting. And I have to say, many of the people that are selling this have no idea what they're doing. Okay, I'm done. And I kind of summarized all this, and this is an article I wrote for a trade journal here in the United States about mycorrhizal fungi. And I'll email this to anybody if you want it. And I'll stop there, and we'll see if everybody's still with us, and if we want to ask any more questions. We are actually at 2 hours now. Sorry that I used up all the time.
We'll try to keep it short because we know you're very tired. So,
I'm okay, actually.
Yeah, you okay? Great. It's good to know.
I can maybe use a beer, though. So if anyone has questions, let's see. Okay, there is a question here about low soil in the Negev desert. Am I saying that right? Negev?
Yeah.
I would suspect that that's fairly low P soil, but I'm not positive because it probably depends on the source of the material that blew in. The low soils that are in eastern Washington, they're fairly low P. So I would think the mycorrhizae are going to be pretty important. Of course, you have to irrigate there. I know that. I think one question I would have is there any plant cover at all on the ground in that region that's going to support the mycorrhizal fungi to begin with? Knowing that would be really critical. I'm guessing there's very little plant cover, so that's the one place you probably would inoculate, and you just got to find a good supply of a product that's actually good. And it's unfortunate, but there's just a lot of snake oil people out there. Great question. How do you know how much AMF you have in the soil? Yeah, that's the big problem. You don't, and it's really hard to know. There's actually a place in Oregon that will test it for you, but you wouldn't be able to mail your soil samples here. It's really hard. Only labs can really do this. It would be a really interesting service. And I will say the lab that does do the testing here, they're doing a lot of other stuff. If they try to make a business just out of that, they would not survive. And there's just not enough business. But it's hard. I mean, basically, you have to run a bioassay. Like you can't, it would be nice, and someone maybe someday will develop some diagnostic tools. It could be based on DNA and PCR, for example. Like you could extract a soil and just look for the markers for mycorrhizal fungi. That'd be one way to develop a test. The problem is you need to develop that test and then have the bioassay that proves that your test is accurate, and no one's done that yet, as far as I know. And the only way you can see these guys is, as far as being in the roots, is you have to clear and stain the roots. It is doable. I mean, a winery lab could do it. And there's a couple of shortcut methods that use more common materials. And if anyone's really interested, I'd be happy to share that with you. Basically, there's a method you can use with ink and vinegar. It's not as good, and you don't get nice pictures like I get, but it is possible to stain roots yourself. And really, that's pretty much what you got to do, unless there's a company that's willing to do that service locally for you. Okay. That looks like the last one in the chat box.
Well, does the fungicide sprays that we use on the canopy, do they have effect on the mycorrhizal status in the soil? Like if you spray copper or
Right, that's a good question. I would say most of the fungicides we use on the canopy are not that big of a deal. And the reason I can say that is because I've done the surveys here and I've done them in Washington. We're using plenty of fungicides. The roots still have mycorrhizae on them in good amount. So I don't think it's a huge concern. I think, I'm trying to remember the name of the chemical. There's one chemical, sorry, I'm forgetting the name. It's mobile in the plant, and it's supposedly kills fungi, kills insects, kills a lot of stuff. I forget the name of the chemical, but that might be something we need to look at, and I don't have information on that, but it's the only one I know. Most of the fungicides we're using, even if they're considered mobile, they usually don't go to the roots, right? The one class of fungicides that are a problem are the phosphite fungicides, which sounds weird, but there's a group of fungicides which are made on PO3, not PO4, and those will inhibit mycorrhizae. I actually had a slide in my early version of the talk that I took out that showed that, but so I don't know that anybody would be using that in a vineyard system. It's normally used to kill oomycete fungi, and mainly in the nursery industry. So the fungicides Alliette, for example, I don't know if that rings a bell, and metalaxyl, those are both phosphite or phosphonates. Phosphonates are bad. And really, they're not bad because they probably are directly toxic to the mycorrhizae so much as they fool the plant into thinking it has a high phosphorus level. And so then it'll downregulate mycorrhizal colonization.
anyone else? Is there something? If not, we will thank you from the bottom of our heart for this very interesting talk in this very late hour.
You're welcome. It was fun.
Appreciated. And
I hope.
Listen to me. I hope everyone assume.
Thank you very much for the talk.
Over the presentation.
You're welcome.
Thank you very
You're
much.
welcome.
Thank you very much.
Thank you so much.
And you're
I will
more than
send-
welcome to visit us after COVID is
Just go over it
Yeah.
because I went
Kind of.
over it, but I want you to, you know, if there's something maybe I didn't.
Thank you very much.
Thank you. Noah, I will email you the PDF of this talk, but it'll be in a couple days because it's just a PowerPoint right now, and I'll have to, you know, get rid of some of the animation and stuff. But yeah, I have no problem sharing that.
Thank you very much. And also the article you showed about how to encourage mycorrhiza.
Yeah, I'll attach that too.
Thank you very much.
All right.
Have a great
Well,
night.
I'm ready to go to bed.
Have a good sleep.
Thank you.
Good night,
Bye.
Paul.
Bye, everyone.